Welcome to the CSIR NET lecture series. In this particular series, we'll talk about key concepts, detailed analytics, we'll discuss previous year questions and practice more questions on the similar topic. This lecture is about fluorescence microscopy, and this comes under the methods in biology section in Unit 13. So let's look at the analytics in one go. So the topic this particular topic is included in CSIR Unit 13. The questions are asked in part B and C. The difficulty level is easy to moderate, and the question types that are asked are based on application-oriented stuff and principle of the particular microscope. So in this video, we'll talk about fluorescence microscopy, which uses the principle of fluorescence for imaging specimens. We'll learn this concept in 20 minutes. We'll learn the principles of fluorescence, working principles of fluorescence microscopy, applications of fluorescence microscopy, and the limitations of fluorescence microscopy. This video is in English version. If you want a Hindi version, you can click on the I button to get that. Anyway, the application and the limitation part is super important from an exam point of view. Before we start, let me tell you the resources that you can refer to. First of all, the generic resources Pathfinder books are good. So there is a book called Biophysics and Molecular Biology in the entire book set, from where you can read and revise this chapter. Anyway, if you have more time, you can also refer to Wilson and Walker. Generally, people do that if they have time. After this video, you won't need any of these resources. So let's talk about what is fluorescence. Fluorescence occurs when an excited molecule, atom, or nanostructure relaxes to a lower energy state, usually to the ground state, through emission of photon without the change in electron spin. This is very important, without a change in electron spin. Now let me try to explain this in a diagram. So here you can see some particular atom or particular molecule is in ground state. With a particular excitation light, it gets into an excited state, then gets back to an ground state again, and in this process they emit particular energy, and that energy corresponds to a particular wavelength, and that's what we see as a fluorescence. And this phenomena happens in order of 10 to the power 9 to 10 to the power 7 seconds. In order to understand fluorescence in a better fashion, we can use a Jablonski diagram. In the Jablonski diagram, we can see the ground state is S0. So here our excitation light in a blue wavelength, let's say, excites a fluorescent molecule. So it jumps into an excited state and then relaxes and come back to the ground state again. And in this process, it emits fluorescent light. Notice that this particular case, the fluorescence light is in green wavelength. That means it has shifted the wavelength. So in fluorescence, two important thing is the wavelength shift of the emitted light, and the spin of the electron remains unchanged. And it's a fairly fast phenomena happening in order of 10 to the power minus 9 to 10 to the power minus 7 seconds. So this principle of fluorescence is used in the fluorescence microscopy. So let us look at the light path of the fluorescence microscopy in bit more details to understand that. But before that, we need to start with a fluorescent specimen. Otherwise, how would we visualize? So we can use immunohistochemistry to label some of our specimen. Maybe we can label some particular protein with immunohistochemistry. If you want to learn more about immunohistochemistry, the link would be in the I button. So obviously in this case, we can see the fluorescence is coming from the nucleus. So possibly we have labeled a fluorescent protein which is present in nucleus. Otherwise, we can also express our particular protein of interest with the expression vector like this, and then track the fluorescence with the microscopy. In this case, it's also from the nucleus. That means this particular protein that we have labeled with GFP is in the nucleus. So let's look at the light path. Now imagine we have a fluorescent sample in this plate. So here are the arrangement of the optical elements. You can see the sample here, objective, the filter cube, excitation light, and the detector. Detector is mostly camera. So first the excitation light hit the filter cube, and it is diverted through the objective and falls into the specimen. Now, obviously, when it falls into the specimen, it excites the fluorescent molecules present in the specimen, and it jumps into an excited state. Then it relaxes and comes back into a ground state, and in this process, it emits fluorescence. So you can see the fluorescence light passes through the objective and travels towards the detector. And in the Jablonski diagram, you can see the same. The similar process can be visualized in terms of intensity profile. So this is the intensity profile of our excited or excitation wavelength. And this is the intensity profile for our emitted wavelength. So already we can see that the wavelength is shifted. The wavelength that is seen as fluorescence is at higher wavelength, right? That means lower energy. Higher the wavelength, lower is the energy. This phenomena is known as Stokes shift. So Stokes shift is an important terminology that can be asked in the examination. Let us look at this filter cube in bit more details. It has an excitation filter, dichroic mirror, and an emission filter. The filter type could be different, such as band pass filter, low pass, or high pass filter. Low pass means it allows a lower wavelength to pass, high pass means it allows a higher wavelength to pass, and band pass means it allows only a restricted band of light to pass through these filters. Anyway, this is beyond the scope of discussion for this particular video. Anyway, let's get back to a microscope and try to look at the light path in a cross section of a real microscope. So here you can see the objective lens, camera, and the lamp house. Now we are going to look at the light path in the same diagram. So first of all, the most important component is the filter wheel, which has all these filter cubes. Filter cubes for different wavelengths are comprising this particular filter wheel. So the light comes from the lamp source and it goes through the filter wheel, pass through the objective because it is diverted through the dichroic mirror, and falls into the sample which is kept here. And the fluorescence light pass through the objective itself. Now by changing the filter wheel, you can just change the illumination wavelength. For example, previously we were illuminating with a blue light, now we are illuminating with a green light, now we are illuminating with a red light. So just by changing the wheel, we can determine that which wavelength we want to illuminate the specimen with. And it depends on what type of fluorophore we have in our specimen. Okay, in this case we have a GFP specimen, so we have illuminated the specimen with a wavelength of like some around 488 nanometers in the blue wavelength. And obviously the fluorescence would be emitted and passed through the objective lens. Then with the combination of mirror, it gets deflected and it can pass through specific prisms and array of lenses. Either it can come to the detector, that means in the camera to be detected, or it could be diverted towards the eyepiece as well. And we can see the fluorescent specimen with our eyes. So both are possible. By changing a knob or pushing a mirror in the path, these pathways can be changed. That means whether it goes to the camera or whether it goes to the eyepiece. And ultimately, we see or we image these fluorescently labeled cells. In this case, there are some cells which are labeled with fluorescence. So now we understand the key part of this fluorescence microscope is this filter cube, because by changing it, we can change different wavelengths of excitation, right? Now let's talk about the application of fluorescence microscopy. So fluorescence microscopy can be used in biomedical research, or it has clinical applications as well. So let's talk about the biomedical research part. We can use fluorescence microscopy to study subcellular localization of a protein. So in this case, we have immunostained, or might be we have tagged a protein with GFP and visualize the protein localization. First example shows the protein is localized in the membrane. That's why we see the fluorescence from the membrane. In second example, we can see the protein is localized in the nucleus. And in third example, we can see the protein is localized in the mitochondria. So overall, we can look at the localization of the protein in the cell. So that's application number one. Okay, application number 2, we can visualize specimen in real time under fluorescence microscope. This is called live imaging. We can check several cellular or organelle dynamics. For example, if we want to look at the mitochondrial dynamics, the fission and fusion event of mitochondria, we can use this kind of microscopy technique. So in this case, we label the mitochondria with some kind of dyes or some kind of protein which expresses a fluorescence protein, and we can see the dynamics in real time. We can also track the migration of a cell using fluorescence microscopy. So we just have to label the cell with a fluorescent dye like this, and we can see how this cell is moving from one part of the tissue to another part of the tissue. And this is done in live imaging. That means the cells are alive and we can see their motions, see their movement in real time. So it's another application of fluorescence microscope. So already you can understand we can look at live specimen using fluorescence microscopy. In neurobiology, scientists used fluorescence microscopy to understand how neurotransmitter containing vesicles are reaching the synapse. It is produced in the cell body and now it has to reach to the synapse, right? And in this road, they have to be passed from the cell body to the synapse using these small molecular motors known as kinesins or dyneins. So this motion of vesicles can be detected using fluorescence microscopy. For example, scientists can quantify the movement time scale, the movement kinetics, the movement direction, whether the vesicles are moving in an anterograde direction or a retrograde direction. So all these can be done using fluorescence microscopy. Now, application number 5, we can also monitor the activity of neurons simultaneously using a time-lapse fluorescence microscopy. So we have to express a genetically encoded calcium sensor, or let's say a calcium sensitive dye in a neuron, and we can monitor the neuronal activity from many neurons simultaneously. Now, disadvantages of fluorescence microscopy is also important to understand. So first of all, fluorescence microscopy gives a blurry and out of focus image. Now, photo bleaching of the specimen is another problem, and the cells under investigation may themselves be damaged in the process of fluorescence microscopy due to phototoxicity. Now, poor signal to noise ratio is due to higher background. So generally in the left hand side image, this is your expected image, but due to a high background, you see an image like this, much blurry and a lot of background in this image. So confocal microscopy is one variant of fluorescence microscopy that can take care of all these problems. In next video, we'll talk about the confocal microscopy and how these problems are overcome. Now, photo bleaching is the biggest disadvantage of fluorescence microscopy. So due to photo bleaching, the fluorescence dies off like this. Okay, let's talk about the resolution part. So the resolution is defined by this equation, d equal to 0.61 lambda by Na, numerical aperture of the objective, or mu sine alpha. So here, what does alpha mean? So this is the illumination light or the excitation light. Here alpha is the half angle with the optical axis, and the lambda is the wavelength, and mu is the refractive index of the immersion media. So obviously, we can have different scenarios. Like D is the minimum distance between 2 points that can be possibly resolved. We can either have situations where two things are nicely resolved. We can also have some situation where the things are just resolved, which is known as Rayleigh limit. And also we can have situations where the things are not resolved at all. So these are important aspect of the overall microscope resolution. Now in wide field fluorescence microscopy, the whole specimen is illuminated at one particular point of time. That means here we are imaging and focusing the microscope in one plane. So obviously we expect fluorescence molecule in that particular plane should be illuminated. But what happens is this is a wide field illumination. That is why the plane above that or below that or below the plane of focus or above the plane of focus also gets excited, and they emit fluorescence. And when we collect the light, we collect all of them. That leads to out of focus blurry images. And this problem is taken care of in confocal microscopy. Okay, anyway, let's look at the application of immunofluorescence in clinical application. Like we have already looked at the biomedical research part, we can also look at the clinical application. So for several diagnostic tests, these fluorescence microscopy based techniques can be used. For example, in one variant of ANA test or anti-nuclear antigen test, looking at the fluorescence patterns of autoantibodies would really help. It's an example of indirect immunofluorescence. Also, diseases like bullous pemphigoid, then pemphigus vulgaris, which are like skin diseases, can be diagnosed using immunofluorescence based approach. Now let's go to the exam corner and try to understand what are the previous year questions, and let's solve them. So this is a question from June 18. Here the question says, conformation dynamics can be determined using which of the following technique? Fluorescence microscopy, NMR spectroscopy, mass spectroscopy, and differential scanning calorimetry. So you can pause this video and try to answer. If you have answered correctly, the correct answer is fluorescence microscopy. Fluorescence microscopy can highlight intracellular structures, cellular dynamics, or organelle dynamics. It cannot show us about molecular dynamics in details. Question number 2 is from December 2018. In this question, they have asked, in order to visualize the intracellular organization of a cell, one can utilize various microscopy-based techniques like differential interference contrast, phase contrast microscopy, dark field microscopy, epifluorescence microscopy, scanning electron microscopy, transmission electron microscopy, confocal microscopy, etc. Now, question is, which of these techniques can be used to study intracellular dynamics using live cell imaging? That means you have to identify those techniques which would be appropriate for studying live cell. So obviously you have to use light microscopy for studying live cell, right? And the correct answer would be B. That means all these differential interference contrast, phase contrast, dark field, all of these are light microscopy variant. So these are bright field microscopy. And also the epifluorescence and the confocal uses the fluorescence to image the specimen. So again, it uses specific wavelength of light. So you can see this particular question was asked in part C. So this question was easy, directly application oriented. And if you have understood the lecture, you can easily answer these kind of questions. Now let's practice some CSIR style MCQ questions. Question number 1, which of the following is not an application of fluorescence microscopy? So obviously the questions are asked from an application point of view. So this would be useful for that. So the options are subcellular localization of a protein, tracking migration of a labeled cell, analyzing rate of transcription, analyzing ultrastructure of biomolecules inside the cell. Correct answer is option D, because analyzing ultrastructure of biomolecules can be done using NMR, mass spec, maybe all the X-ray crystallography, these kind of biophysical technique. We cannot use fluorescence microscopy to do that. Question number 2. Which of the following statement about fluorescence microscope is wrong? It can be used to study biological specimens. Mercury arc lamp is used in the scope as a light source. The image in the microscopy technique are always sharp and never blurry. Emitted fluorescence light has higher wavelength than the excited light. And the correct answer is option C. You already understand that the biggest disadvantage of fluorescence microscopy is it has a blurry images. So that is why you can understand option C is the correct option. Which of the following microscopy technique does not use the principle of fluorescence? Confocal microscopy, two-photon microscopy, FRAP, or DIC microscopy? Correct answer is DIC. Differential interference contrast microscopy does not use fluorescence. It uses different principle to highlight unstained specimen. Anyway, as per summary, we can quickly see what we have learned right now. We have looked at the light path of the microscope in details. We looked at the Jablonski diagram to understand the Stokes shift and the overall principle of fluorescence, and we also looked at the application. You can get these kind of summary diagrams in my Facebook page. So obviously check out the Facebook page and Instagram page. All the links are provided in the description. You can support this channel using Patreon, Bhim UPI app, or by clicking on the super thanks option in the bottom right corner of any video. You can contribute using Paytm, PayPal, UPI. The contribution starts from rupees 40 and it goes till rupees 1000. Your small contribution means a lot for me. Anyway, you can follow my other channel, Animated Biology with Arpan, and in order to get exclusive medical related content, you can follow the Nerd Medic channel. As usual, don't forget to like, share and subscribe. Do let me know in the comment whether you like this video, if you want any modifications, or if you want any attachment to these videos. Your feedback would be very important to make these lectures better. So see you in next video.